Hello everyone. Today I'm going to talk about evolution through molecular comparisons. We can learn about evolution. We can learn about evolutionary relationship between different organisms through molecular comparisons. Like when we know about, or if we know about the molecular anatomy of different organisms or different species, we can know about the evolutionary relationship between those species, like when those species got diverged from a common progenitor, what are the relationship between these species. We can know about evolution through molecular comparison by comparing the molecular anatomy of different organisms. So this is one I'm going to explain in this lecture. So let's proceed. Complete genome sequencing of number of organisms have been done. Okay, complete genome sequencing. That means number of organisms, genome. We have the complete information about the genome. We know the complete sequencing of the genome of certain organisms, like bacteria, number of bacteria, the genome of number of bacteria have been sequenced. Then many archaea, a few yeast species, some of the plants, a roundworm called C. elegans. Then there is Drosophila, mouse, rat, chimpanzee, and human beings. So we know the complete genome sequencing of all these organisms. So, a detailed and quantitative comparisons of sequences provide deep insight into the evolutionary process. Now we know about the complete sequence of the genomes of different organisms. By detailed and quantitative comparisons of these sequences, we can know about the evolutionary process that these organisms have gone through. Now, first of all, we should know about different kinds of sequences that are there. So we have mainly three kinds of sequences. One is homologous sequence, the other is paralogous sequence, and the third one is orthologous sequence. The homologous sequence is when the two genes share readily detectable sequence similarities. When the two genes are have sequence similarities, when the two genes are similar in sequences. They are not identical, but they are similar in sequences. Then these sequences are, or these genes are called homologous, and the proteins they encode are called homologs. Homologs. Okay. The second kind of sequences is paralogous sequence. When the two homologous genes occur in the same species, okay, when the two homologous genes occur in the same species, they are called paralogous, and the protein they encode are called paralogs. So these paralogous genes are presumed to have been derived by gene duplication followed by gradual changes in the sequence of both copies. So suppose there was one gene which was accidentally duplicated into the genome, so the two genes they become paralogous. Slowly, the two genes they undergo a little mutation. Okay, so gradual changes in the sequence of the both genes took place. So the two genes they are similar in sequences, but they may have different functions. Okay, so this is about paralogous sequence. Then third one is orthologous sequence. So, two homologous genes or proteins they are found in different species. Then they are said to be orthologous. Okay, the two homologous genes, that means the two genes they have sequence similarities, but they are present in different species. So they are called orthologous, and the protein that they form are called orthologs. So orthologs are commonly found to have same function in both the organisms. Okay, for example, genes which are responsible for synthesis of hemoglobin. Okay, all animals they have hemoglobin, they form hemoglobin in their body. So the genes which form hemoglobin they are called orthologs. These genes are there in all the animals, but they have a sequence similarity, and but all the genes, they play a same role. They all form hemoglobin, which play a same function. So this is about different kinds of sequences. So all the genomic data, the sequences of the genomes uh, of various organisms, they are stored into a genome data bank. So whenever a new genome is sequenced, uh, the newly sequenced genome is compared with the genome sequences which are there in the genome data bank. So a newly sequenced gene, a newly sequenced gene, when found to have a strong similarity with the orthologous gene, its product is presumed to play the same function. Okay, when a newly sequenced gene, when it is compared with the genome data bank that we have, and we found to have a strong similarity with the orthologous gene. Okay, so we can presume it can be presumed that the newly sequenced gene play the same function. Okay, so let us understand this by the example. Suppose the orthologous gene is a hexokinase gene, a gene which produces hexokinase. Okay, and the newly sequenced gene has a strong similarity with the this gene which produces hexokinase. Okay, suppose the newly sequenced gene has a 80% similarity with the gene which produces hexokinase. So we can presume that the newly sequenced gene produces hexokinase and have the same function. So therefore, by knowing the sequence of a gene, we can deduce its function. Moving further, so what is an annotated genome? Annotated genome is basically a genome. We know the sequence of the genome, but along with that, we also know about the likely functions of the gene product. Days we have annotated genomes, and we know about the gene sequence of. We not only know about the gene sequence of the genome, we also know about the functions of the gene products. So the sequence differences between homologous genes may be taken as a rough measure of the degree to which the two species have diverged during evolution, of how long ago their common evolutionary precursor gave rise to two lines of different evolutionary fates. The large the number of sequence differences, the earlier the divergence in evolutionary history. So, by knowing about the sequence differences between the homologous genes, whether the difference is like 30%, 40%, 50, 60%, like 90%, by knowing about the differences between the homologous genes, we can have a rough idea that to which degree these two species have diverged during evolution, how long the two species diverge from a common evolutionary precursor, and so more the number of sequence differences, more the difference, earlier the divergence in the evolutionary history. So, see, as organisms evolved, they acquired new structures, new processes, regulatory mechanisms. All these are reflected in their genomes. Okay. For examples, if we compare prokaryotes with eukaryotes, prokaryotes, these are simpler organisms. They do not have genes for intracellular membranes, but eukaryotes, they have genes for the intracellular membranes, because prokaryotes do not have intracellular membranes, but eukaryotes have intracellular membranes. So during evolution, eukaryotes evolved to acquire the intracellular membranes. So they acquired the genes which are responsible for formation of intracellular membranes. Likewise, if you look at mammals and invertebrates, mammals have genes for placenta formation, whereas invertebrates do not. So mammals, while evolving, acquired genes for placenta formation, because they have placenta, but invertebrates, they do not have placenta, so they do not have genes for that. So all as the organism evolved, um, they acquired new properties, the new sophisticated, complicated properties which are reflected in their genomes. So, coming over to functional genomics. So, functional genomics, what is functional genomics? It deals with studying the expression and function of genome. Okay, functional genomics deals with studying the expression and function of genome. So we have molecular geneticists. Okay, they group genes according to the processes they are involved in, and thus find what fraction of genome is allocated to each of cells activities. Okay, so it is the duty of a molecular geneticist. They actually group the genes. They know about the genome sequence, and they also know about the function of the genes. So, what they do, they group the genes according to the function or the processes that they are involved in, and they find what fraction of the genome is allocated to each of cells activities. Like, for example, genes in E. coli or Arabinose thaliana and Homo sapiens, they consist of genes of unknown function which make up to more than 40% of the genes in each species. So if you look at E. coli, uh, Thaliana, A. thaliana, and Homo sapiens, 40% of the genes have unknown function. Okay, 40% of the gene have unknown function. So, other example is that the transporters that move ions and small molecules across the plasma membrane, they take up a significant proportion of the genes in all three species, more in bacteria and plant than in mammals. Okay, so there is a fraction of genome which is involved in transportation of ions and small molecules across the plasma membrane. Okay, there is a fraction of genome which is involved in transportation of ions and small molecules across the plasma membrane, and that fraction is like 10% in the E. coli, 8% in Arabinose thaliana, and 4% in Homo sapiens. So, molecular geneticists, they have actually done this. Okay, molecular geneticists, they have grouped the genes according to the processes that they are involved in, and they have found out what fraction of the genome is involved in what kind of process, in different organisms. So, in general, more complex organism, more proportion of the genome is dedicated to regulation of these processes, and less is used for basic processes. Okay, more complex the organism, more proportion of the genome is dedicated to regulation of the processes rather than the basic processes. For example, Arabidopsis thaliana, in Arabidopsis thaliana, 2% of the genome is dedicated to protein synthesis, whereas 6% of the genome is required for targeting proteins to their cellular locations. Okay, so less genome is dedicated to the basic process like protein synthesis, and more genome is dedicated to process like complex process like targeting protein to their cellular locations. Okay, so more complex the organism, more proportion of genome is dedicated to the regulation of the different complicated processes than the basic processes. So you see how functional genomics is helping us know about the functional and evolutionary relationship between different organisms. So isn't it awesome? So I hope you liked the lecture, and thank you for listening to me. Take care and bye bye.